critique and suggestions of how we can do things better and how we can work with you. So we also hope that there's plenty of time. In fact, we've allowed plenty of time for discussion, for you to tell us what you're doing that might be pertinent or related, and how we can work better together. So we have 2 seminars. The focus, I'm sure you're aware, since you've chosen this because you're interested in our wheat activities. So we're going to have a presentation first from Carolina Sansaloni about the more genomic, the more genetic analysis of diversity that we've been conducting within the Masagro Biodiversidad Seeds of Discovery projects. And that will, after some discussion of that, we'll then have a presentation from Siva telling us about the pre-breeding activities within the Seeds of Discovery and Masagro Biodiversidad work. So I'm not going to take any more time. I'm going to hand over to Carolina to talk to us about the genomics. Please come in. Welcome, everyone. Thanks again for your attendance, and let's have a great discussion.
Thanks, Kevin. Well, good morning, everyone. I will present today the study that we are doing with the diversity in seed of discovery. And I will start quite quickly with this. It's very well known that all the challenges we are facing, we heard many times, all the challenge to food security and nutrition. So the breeder require new genetic variation to improve some traits. So the gene banks are a very good reserve of all these variation. We know that some are valuable, but variation, other neutral, but most probably deleterious. So the objective is to identify the accession and novel allelic variation to improve the varieties. But it's quite hard if we don't have any information, only passport data. If we imagine, if we need to phenotype the whole gene bank, it will be impossible. It will take a lot of time and, of course, a lot of resources. So genotyping is an efficient and unbiased and cost-effective method for the characterization of the gene bank, especially a large gene bank like CIMMYT. So the objective of this study was to evaluate the genetic diversity among wheat germplasm group between CIMMYT and also Icarda accession, including wild relative, land races, and elite breeding line, to see what diversity is underexplored in both gene banks. So what we did basically was to genotype almost 100,000 accession from CIMMYT and Icarda gene bank, 32 accession coming from Icarda, and the rest is from CIMMYT. We divided in 3 main groups: the hexaploid group, tetraploid group, and wild relatives. We use Darcy technology for the characterization. We choose this technology because avoid ascertainment bias, because it's not based on one reference genome, so we can explore more diversity and exotic alleles. Well, as I say, we divided in 3 groups. We align all the markers on the last version of the reference genomes, and this is just an example. One small part basically is the chromosome 1, 2, and 3 in the genome A, B, and D. And here you can see the distribution. In green are the number of gene models per bean. We divided all markers in beans of 10 megabases. And in here you can see how many markers. So it's a spective shape. It's always look like a U, because most of the markers and genes are on the size of the chromosomes, and in the middle are the centromeres. So in blue you can see the number of Darcy markers, and in orange the number of markers overlapping with genes. So it's quite nice, the distribution of the markers and genes. In this picture you can see then the percentage of markers aligning uniquely on the genome, and it's quite nice. This is the hexaploid group, which is expected that most of them, more than 75%, align uniquely on the genome. Of course, it's also expected that the wild relative, it's not pointed anyway. The wild relative only 50% align. We need to consider that in the wild relative group we have 27 different exotic species and more than 11 different genomes. So it makes sense that are not even A, B, and D. In the tetraploid, we align in both, in the Chinese Spring reference genome and also in the SVEVO, which is the tetraploid reference. And this is the first result. This is the hexaploid diversity analysis, in which we include 60,000 accessions from 8 species, but mainly Stibun, bread wheat. All these accessions come from 105 different countries. And these are the number of markers we use. But it's unfortunate that it's not, okay, I go here. So what you can see here are in blue, the land races, in red, the lead material, and in green, all synthetic material. And it's nice to see, well, when it turns, it's easy to see that there are few areas of blue, of land races, that are not sampled by the lead material. So are areas that we believe that are underexplored by the modern germplasms. And then all the lead material tend to be going one corner, so there is a bottleneck in diversity. But after that, new diversity is created with the synthetics. So it's quite nice to see this. Then we can discuss more. So the same picture, exactly the same picture. What we did is a hierarchical clustering analysis, in which is based in modified Roger distance. So it's exactly the same picture. But what we did is we split all accession in two groups, in two clusters, based on genetics, diversity. And then the group, in this case, is split in two. The first one, we think that are more original germplasms or old germplasms, and modern germplasms. Then the group with more diversity is split again in two, maximizing the diversity between groups and minimizing the diversity within groups. So then, and it's created another two groups. So the modern germplasms divide in synthetics, and a second group which mainly are elite and new and more modern land races. And then the process continue, splitting in two clusters in each level. And we calculated also FST values. And the FST values give us an idea of which level of cluster explain better the diversity. In this case, the biggest values are in cluster 6 and 12. So we focus on 12, and we can see in blue, all these blue are Asian land races and a little bit of Mexican land races. So mainly from Iran, Pakistan, Turkey, China. And then the other group, more in red, are the lead material. And this is quite interesting because since cluster 5, the block of elite keep the same. And never until cluster 30 something, never split. So this is quite an interesting result. And then Mexican land races that start from here and divide into a subgroup of Mexican land races. And here we can see the synthetics. And a very little part, we identify these as outliers, and I will show you why. I will show you later. So this is exactly the same clustering analysis, and in the first separation, we look at the genome level. So these are all markers. Actually, these pictures show each chromosome, chromosome 1A, 1B, 1D, 2A, 2B, 2D, and so on. So what we wanted to see here is the FST values per chromosome to give an idea which chromosomes are involved or are driving the differentiation of these two clusters. And here we can see that mainly chromosomes A and B are driving this differentiation. But in the second division, it's interesting to see that the synthetic group is separated here, and all D genome are involved in this differentiation, but which, of course, because we are including an exotic D genome. So this is again the same, but in a different perspective. In here you can see each cluster, so cluster 2, 3, 4, 5, and so on until 12. Only the chromosome, just an example, we have this for all chromosomes and all clusters, but it's just an example. This is a group, the chromosome 3D, and it's again the same than before, the same than this, but looking at one chromosome. So all this is the chromosome 3D in the cluster 2, in the cluster 3, and so on. So you can see in cluster 3, these are the FSTs per marker basis. So each dot is one SNP, and the ones that you can see in red is because are in a certain level that we believe that are significant. So all red dots are significant, and in this case is the cluster 3, in which the synthetic are separated. So, of course, this is 3D, so it makes sense that the D genome is involved in this differentiation. So we can see in cluster 3 and in 7. In 7 as well, the second group of synthetic is separated from the cluster. So in both cases, the D genome this was a proof of concept that really we can identify some important SNP that are driving all this differentiation. But then we were curious and we went in more detail. Again, this is an example. We align genes on the same picture than before. We wanted to position some important genes for important traits. So this is only one example in which we align post-harvest sprouting in chromosome 3A. These are the same picture. This is the whole genome, and this is a zoom in where the gene is positioned. So we can see that, of course, again, this is an analysis of the FST values on variant per variant basis. So a SNP, each SNP has one FST values, and reveal positive selection, highlight positive selection. So for each, I put here for each cluster split, the highest FST reveal the variant that contribute to the separation of two subpopulations, which may identify molecular footprints associated to potential selective sweeps. So this analysis also identifies genomic regions which are known to be associated with key agronomical traits. And one important thing that we believe is now we can see allele target for future breeding. Why? Because, for example, in here, in post-harvest sprout, this is the cluster 2. Remember when the original germoplants divide from mother germoplants? And we can see high values of SNPs here around the gene. But then, in another separation, when the elite split from modern land races, this disappeared here. So we still have diversity in the original germoplants, in the original land races, that are not present at all in the modern land races or even in the elite material. So we have two options. We don't know, and I will have your opinion. Probably this is really good diversity that was left behind in the selection of the elite material, or probably are not good diversity, and that's why it was selected. But this is some area that we need to continue and we explore more in detail, because definitely we have a lot of exotic alleles that are not used at all in the modern germoplants.
Carolina?
Yeah.
Just a quick question. How do these two figures, how are they related to one another? What's the difference between the figure on the left and the figure on the right?
This is just a zoom in. This is a zoom in which showing the position of the gene.
So some of these little red dots on the right are over there.
Yeah. This is a whole chromosome. And this is just a little piece of the chromosome just to identify the genes.
So where are those red dots on the right?
Here.
On the left.
Yeah.
Okay, so they're in that area.
Yeah,
Okay.
sorry, I forgot to put. Yeah, it's in here. So the second picture is only a zoom in to visualize
So all the dots that are red in the left hand are not all post-harvest sprouting genes.
No. All these red
Yeah.
are significant in the separation of the cluster 2.
Okay.
All germoplants, modern germoplants, all these are significant in the chromosome 3A. Okay? We have for all chromosomes. This is only an example. And these are responsible for the separation of cluster 4, in which the elite separate from modern land races.
So you have any ideas about that little single red dot over there on the right? Do you have any ideas of what gene that is?
No, I mean, we can search. The idea of showing this is that we are creating a great resource, but because we can explore in so many different ways, and each of you can explore in your research in a different way. So we can focus in few examples, but not in all. But definitely, if someone is interested in this part of the genome, in the chromosome 3A, can go in detail and look what is in there. All the data is available. We know each SNP. We also know if the SNP align on different genes. So all this information, it's available, and it's a resource, basically, for continued exploration. So only this is a zoom in of this part. But yeah, I just wanted to concentrate in this area to really show you what we have significance region, and in here there are many SNP. In here you can see only one dot, but there are few dots in the same. So few SNP here, but non-SNP here. So these SNP were not selected in the modern germoplants. Also, as part of this study, we did a GWAS analysis with Carlos Guzman and Wiwi in using we did with few traits, but I'm showing only 2, the grain protein content and sedimentation. And it was a way to show how breeders can use all this diversity in their breeding programs. So in here we are showing only 2 quality traits. We use only almost 4,000 samples that Carlos phenotype. We have more data as well, but we just took a small portion of all information for this study. So almost 4,000 phenotypes for grain, protein content, and also segmentation. For the first trait, we found 18 genomic regions in 12 chromosomes. You can see in here. And in the second one, it was found 19 genomic regions in 4 chromosomes. All, it was quite nice because are all from land races, and we can, looking at the bibliography, we could identify many of them already reported. So we were, happy with that. But also we were more happy because we could identify new regions that were not reported before. For example, this one in the chromosome 2, it's apparently is new. So Carlo is very excited to explore more what is inside. And also in the GCP as well, we identify one new QTL. So again, it was a proof of concept. We want to see if we could, with land races, we could identify regions that are already reported, but also if we could identify new areas. In this case, it's quite nice because we could identify some QTLs that are not related with grain yield. So you can select this QTL without compromising yield. And yeah, I think that's it. So similar analysis we did with the tetraploid group, exactly the same approach. You can see in blue all land races, in red elite material, and it's easy to see that there is a bottleneck in the diversity, but still we can see many elite material that are across all land races, so we still keep a lot of diversity.
Carolina, just a quick question. Land races are always tetraploids, is that correct?
Yeah, in this case, yes. Mm-hmm.
Oh, but you can have hexaploid land races?
Yeah.
Oh.
In the previous analysis were also highlighted in
land
blue.
races could be tetraploid or hexaploid?
Yep.
Okay.
Yep.
It's just a dumb, maze-related question that don't know anything about genomes. Okay.
So it was exactly the same approach. Then we did the clustering analysis, and again, it's exactly the same. It was nice to identify a completely different group in here, and also is all this part in blue. These are land races from Ethiopia, which we know that Ethiopia is very rich in diversity. So yeah, it was nice to identify this group quite separate from the rest. We also identify outlier that I will explain in a moment. And then the elite material here, also from the cluster 5, the block keep the same and never split. So all the elite material are consistent in one unique cluster. And then the different land races. So the third group, it was the wild relative. We include almost 4,000 accessions of wilds, including 27 different species and more than 11 different genomes. So you can see it's very small, but you can see all the 27 wild relatives from Aegilope and Triticum. And yeah, basically one of the most important group is this one, which is Aegilope, which is also split in 2 small groups. A and B genome are here. and the rest is G, M, W, U, C, and so on.
But did the triticums hold together kind of as a group?
Yeah.
I can't tell by the colors.
Yeah, you can see in here. Again, the same concept, clustering analysis. And it's interesting I wanted to compare with the origin of wheat. And basically what you can see here is the genome A, Urartu, and the genome B, Speltoides, are here, these two. And genome D is Tausia. But you can also see the whole diversity of exotic wheat, just to see that we still have plenty of diversity to explore. I'm not saying that all are useful for bread wheat or durum wheat, but we still have a lot of diversity and different genome that, of course, can be explored. And I know they are being explored in the synthetic program. So coming back to the outliers, what we did here is we calculate a core set, because of course these are huge number of accession, and it's just impossible to work with so many, 60,000 hexaploids. So we selected a core set of hexaploid, a core set of tetraploid, and a core set of wild. We recall all together, and this is how it looks. In red are all hexaploid, in green tetraploid, and in blue wild relative. So this is kind of a picture of the global diversity. But it was quite curious to see this is, the color are based on passport information. So all red are hexaploid in passport. All green are tetraploid and blue wild. But it was interesting to see some red dot here and some green dot here and red here. So we say something is wrong. We went to the genome and we wanted to identify all markers belong to each genome. So what we analyze in here, only we are observing the hexaploid. So the black dot, just forget. We are only looking at in red, the number is less than 10% of the genome markers. So all accessions with red dot has less than 10% of the genome markers. And in purple, more than 10% of the genome markers, means these are really real hexaploid, and the red one are not hexaploid at all. So, so of course, they are passport information, mistake in the passport information. So this is an area, an opportunity for improve also the passport data in the gene bank.
Is this data used when you identify these accessions? Is that information going back
Yeah.
to Tom and he corrects it?
Yeah, we are on the process.
Yeah.
Correcting is too strong of a word, because this problem has been identified at other gene banks as well.
No, I know, but
So,
I mean,
but
are you reclassifying
No,
hexaploid to durum? Or
so
you throw it out?
USDA is actually doing chromosome squashes to confirm the number of the ploidy of the materials, because visually it looks like a misclassification, but the only way you can tell is by counting the chromosomes.
To confirm what they come up with then.
Yeah.
But we were doing an analysis with
we
Chio
have
as well, because I gave all the lists of potentially misclassified accession, and they were looking visually, and if not, they were planning to plant, right, and evaluate. The idea is to, before fix the passport, it will be better to regenotype and confirm they are true, and then I think we have all proof to
But also you say
reclassify.
less than 10% D genome markers.
Yep.
Could there be cases in which the D genome is there, but we don't have sufficient coverage of markers?
No, because you will observe in more, and there's are very few. So no, don't think so. No. And Chio already mentioned that she found in some of them that are real durum. Visually, she can see that are durum. So this already, I don't know if you already fixed. Or, yeah, okay, yeah.
I mean, the basic assumption is that what is on the label for these suspicious samples, what is on the label is correct, right? And then you do the investigation to figure out whether which hexaploid, which seems to be a durum, whether it is right. I think in most cases if you come from here and you assume the label is wrong, you can save a lot of work.
Yeah. This is
but
the idea.
It needs to go outside. I mean, Masahiro could count chromosomes perfectly straightforward. Whether you have 28 or 42, that should be
Yeah, but sometimes just by looking at the seed, you can confirm, and they already confirmed in some cases. In the cases that they couldn't, I believe we can regenotype or even plant and confirm. Yeah.
I mean, my point is go with the assumption it's wrongly classified and use the most simple approach. I mean, maybe growing it out in the field, that tells you already it's actually rotum.
Yep.
Before we go all the sophisticated assumption, there are, I don't know, we found a new D genome for which there are no markers.
Yeah. No. So exactly the same, but only looking at the tetraploid, again, exactly the same. We show that in here we have accession with more than 20% of the genome markers. So yeah, of course are hexaploid and not tetraploid. So yeah, we generated a list of this material and we passed to the gene bank. So some of the conclusions, so far in this analysis we include 80,000 gene bank accessions from both gene bank. We identify more than 300,000 markers. 72% align uniquely on the reference genome, but more importantly, 50% are directly linked to genes. In the hexaploid group, we could identify a lead breeding line and synthetic derivative that form a distinct group, and also land races that remain underexplored or unexplored at all. So this is an area that we can continue working and investigating what variation is in there. In the tetraploid group, we assume that breeding programs have skewed representation of the allelic diversity, but still we could identify some elite material across land races or keeping more diversity, which is really good. In the wild relatives, we could identify all species at the genome level very, very well. So this could be an area to explore more as well, identify exclusive markers that could identify a specific species. We have the list of markers belong to different species, so yeah, it can be explored in different ways, no? But the good thing is that the methodology and the technology we use help us to identify at genome level. Then the analysis of FST across the genome highlight areas of positive selections and indicate target for future breeding. As I say, we align different genes. I show you only one, but we have data for different genes in which we can see in each cluster if there is still diversity that were not used in the lead background. And finally, the GWAS analysis identified potential new genomic region controlling these two traits. So grain protein content and sedimentation. And that's it. Well, of course, this was a work a collaboration with many people, not only from CIMMYT, but also from NIAB in UK, from DART in Australia. So it's a group of people that were working together to do this diversity analysis. Thank you. Any questions? Yes.
You can take a few minutes trying to.
Yeah.
Sorry, a great presentation, Carolina. One question. So when you showed the second last slide, are you just looking at markers that align to the bread wheat genome? Or are you, because you're looking across the tetraploids and hexaploids, so the second last slide.
The second last?
The one before conclusions.
I understood the second. Here?
Yeah. So the markers that you use for this analysis, are you just looking at the ones that align to the bread wheat genome?
Yeah. Mm-hmm.
And have you looked at the ones that align to the tetraploid and the D genomes and did this analysis the other way around as well?
Yeah, the problem is with the tetraploid, of course, you don't have D genome, so you can't prove these that are misclassified. Yeah. So it's not really useful because, you know, you are sure that align to the tetraploid, but you are not sure that align or not to the D genome. So for this purpose, we did, but we are not showing here because it was not really.
Was there like too many SNPs in the D genome versus the bread wheat's D genome, or how was that?
No, it's quite similar.
Okay.
The number of markers, yeah. We have a table in which we compare tetraploid in Chinese Spring and tetraploid in SVEVO, and you can see that the number of markers are quite similar. Of course, the difference is in the D genome. Yeah. In Chinese Spring, always align something, very little, but something aligned in the D genome. But it's very little compared with AB. So, of course, we know that it's not true.
Thank you.
Yeah. Sorry.
Carolina, you said for the main protein on chromosome 2, you have this new QTN. And also, if you see GPC, it has ortholog also in chromosome 2B.
Mm-hmm.
Is it the same, or do you find something new?
For the protein content?
Yeah.
No. For the one that we found new is in sedimentation. Yeah, that this one Carlos was quite happy, and he wants to explore more, probably it as well. Yeah. Yes, Maria. Yes.
I'm looking at your second slide.
Okay.
Three plots.
That one? Yeah.
Yeah, I mean, in terms of, a lot of the analysis is like looking at individual QTLs or individual regions. I mean, what this tells us is that there is, I mean, as well, if we are thinking back about the breeding history, I mean, there is an evolution.
Mm-hmm.
Yeah, I mean, things recombine, not randomly. I mean, in terms of, you have linkage blocks. Is there any way that we can establish, like, the evolution of essentially what has recombined well, I mean, like the famous detranslocation, in terms of, based on the data we have, is that methodologically possible? Or as well to say what are successful, I mean, blocks or gene networks based on this? Because, I mean, what this says is that things are not, I mean, they are different from the land races, but they are similarly different, and there is an evolution, yeah? And can we capture that?
Yeah, exactly this we are doing with this approach, because in here, this is not it's not the time, but I mean, I believe that these are original germoplants and these are modern germoplants, but it's not like in a timeline, right? But what we can and what we are trying to explain is where we, or for example, in this case, it's a way to explain evolution and what areas of the genome were selected and what areas of the genome were left in the process of modern material. So we are identifying these areas that were, I'd say, left behind, no, in the selection, and we want to explore what these areas has, and if they are important, maybe are not. Maybe the process of selection works, and you know, and
So what is there the next step then? I mean, in terms of better understanding of why that and not something else. I mean, in terms of what's successful in the selection.
What we are really looking in detail in one of each of these dots, and we are looking at which gene are there, and because we have all the information in the modern and in the old germplasms, so we can go in detail up to the gene level and see not only if it's present or not, but also the allelic frequency of each one. If we still have genes with very high allelic frequency that are not present in the lead material, so this is an area that can be explored more in detail by the breeders, right? So yeah, we are definitely looking at this. Hans, sorry.
I have a very similar question. The landraces, many of the landraces which were used here came out of our gene bank.
Mm-hmm.
And in particular, the Iranian and Mexican landraces and a lot of the Turkish landraces have been used in crosses.
Mm-hmm.
So can you link this data with the PMS and extract the advanced lines which came out of the crosses with the landraces, and many of the landrace-derived elite lines have been recrossed, and then you can check whether some of these alleles were lost during the process, because that would indicate to me they are something undesirable. Some may be still there, and so before we go into huge things here, I think we should extract some of the genotypes which were used for crosses, and then follow up with them and look what happened with these alleles over the last, 20, I mean, some of them have gone through 3/4 crossing cycles now. So you could see whether they were lost. I mean, this is a little bit what the Germans did on their elite lines recently, where they looked over the last 50 years which alleles disappeared and which alleles stayed. And I think this would be a very good starting point to really follow closely up. And maybe we then have to go to some alleles and cross them again. And if the allele is in the first round of elite lines still present and lost later, you don't have to go back to the landrace. That makes the breeders also more. It will attract the breeders more to cross it again.
Yeah, definitely, yes. Of course, we have the identification of each succession and come from the gene bank, so it will be quite easy to grow.
Well, I really think we need to move on, because we have another, I mean, every time I hear this presentation, I find more exciting things. So thank you very much, Carolina. You have at least 3 questions that you need to follow up on one-on-one. It was Hema, Sarah, and who else had a hand up? So at least talk to Daphne? Stephanie. So be sure to talk to Stephanie and Hema and Sarah for your questions. Let's give Carolina one more round of applause. Thank you very much. And I'd now like to invite Siva to join us. Siva has been on this team for about a year, just a little bit over a year. Siva took over from Sukhwinder Singh, who most of us know very well, and has been doing some new things, as well as pursuing following up on some original works by Sukhwinder and other predecessors. So Siva will tell us a little bit about the pre-breeding activities.
This one or this one? Okay. Yeah. Thank you. As Kevin mentioned, I'm with the seeds for about an year. And it's basically, working on the pre-breeding in wheat. And what I'm going to talk is basically about the activities we are doing for the last 1 year and some future plans related to it. And as you know, pre-breeding is basically a bridge between genetic resources and crop improvement. And this slide, I took it from a publication in 2013, which shows, the length of pre-breeding, how long it takes, and especially if it is from genetic resources, and it goes to, varieties in the end by the breeders. And this is a thing I took from the Masagro website, where they conceptualize when you have a good allele, we call it an exotic allele in a different background, which is not the ones breeders normally use. It's not in a working collection. It's not in an elite background. To format it into a background where breeders can readily use. So that's what, that matches the first slide, like what is pre-breeding and how to use it. So in CIMMET, in wheat pre-breeding, we already have a pipeline which is established by Matthew for the last several years, which starts with, land races, synthetics, especially primary synthetics, which Masahiro makes, then figs, collections from ICARDA, and there are trait donors, like certain traits you can find it in land races or synthetics or even semi-elite materials. And then high GE, BV values, that is basically based on genomic prediction or even using markers. And that goes through a crossing section where the crosses are based on, diversity, it's based on pedigree, or is it based on conceptual models in the physiology program? So there is a criteria which goes through the crossing. And once it goes through the crossing, it goes through generation advancement, basically F1 to F6 or F5. And that generation advancement is basically based on yield or based on genetics or genomic selection. And then it finally goes to international nurseries. And in pre-breeding, there is two types of international nurseries. One is called VISIT. It's for high yield potential traits. The second one is satin, and it's for heat or drought. It changes alternate years. So the main job for me was to integrate the results from seed into this pipeline. And we found it's best to integrate those before the international nursery. So we test them, the materials from the seed, test it there, and put it into international nurseries. The other slide looks really simple, but it's really complicated, in terms of logistics, with locations, with different generations, and it's basically a numbers game. Plant breeding is a numbers game, and pre-breeding is also a numbers game. So my main job was to extract the value out of seeds resources, which was generated in the past years. And the first thing I did is to have an inventory of the material. And when I took over last year, it was probably 3 full rooms of materials with seeds, leaf, tissues, and then 2 labs, one in Obregon, one in Bataan, and there was a lot of material. So I wanted to figure out what is happening, what material is there. So the first thing was to make an inventory, and thanks to the technicians, they helped a lot by, doing like 20, 30,000 bottles and putting things together. And we made a list of material that is available, and most of them are related to LTPs. I'll explain in a minute what is an LTP. So there were different LTPs from LTP 1 to 7. It's a linked top cross material. Basically, you take exotics and cross them with an elite, get an F1, cross with another elite, and then do a generation advancement. The same elite, the elite 2 here, is used again in another cross. And then you add an elite 3 there. And it's a chain process. That's why it's called linked top cross. So we have a lot of materials in the linked top cross, LTP 1 to 7. And each one has a different exotics. Like some of them had a synthetic, some of them had land races, some of them were a mix of synthetics and land races. Then there were also materials like Mexican land races, Iranian land races. There are some biparental populations. And there are, rare alleles, haplotypes. There are so many materials there out. And I found out like some of these LTPs, like especially LTP 1 and 2, they were used, they were published also, they were used. And there are LTPs from 4 to 7 that were not used at all. So what we did is we started phenotyping some of the material, and we had several locations. One was Obregon, as you know, the mecca of wheat breeding. Then Toluca, the one for rust, especially the yellow rust. We had also El Batan, it basically used for multiplication and DNA extraction. Then we had also Celaya, where we had a collaborator with whom we worked, and also the Valle de Santiago. So we basically had 5 different locations where we could evaluate the materials. And I also wanted to look at the products of seed, what can we do in terms of using the germplasm and come up with something. And we looked at the past. So in 2018, there was a publication from Sukhveen there where he had identified some contribution of exotic genomes for heat tolerance. And this is, several people in the room was part of that publication. And it basically shows that D genome has a contribution. D genome can contribute to heat stress tolerance. Then, as I mentioned earlier, we have several lines, international nursery, which goes to international nurseries, the visits and satins for yield potential and drought stress and heat stress. So I also put some material in the visits, and we looked, this way goes to probably 40, 50 locations, even though we get requests about 100 locations. We get data maybe half or sometimes lower than that. And this analysis is based on about 15 locations data. And what you see here in the red, is basically the checks. This is the Borlaug, this is the Sokol. And Sokol is one of the, this is actually for yield potential. So, and the blue lines are the ones coming from seed. And these ones are coming from the physiology program. So these are PT lines. This is the line from checks. This is the lines coming from seeds program. And the main purpose of seeds of discovery was for heat and drought, but there are also some useful lines for yield potential. The second one is the satin, and especially when satin is in even numbers, 8 satin, this is for heat stress tolerance. Again, this data comes from about 15, 16 environments. The same coloring pattern. The green ones are physiology lines. The blue one is a seed line. The red is checks. So this is Borlaug. This is a local check, and this is Sokol. And Sokol is one of the very stable line which we use in the physiology program for several years. I also mentioned that we had a collaboration with INIFAP in Celaya, and there it was planted in March to have heat stress conditions. And this is basically the bar plots of all the lines. We sent about 300 lines. And from that results, the local check was here, Sokol and Borlaug was around here. There are certain lines still, with exotic genome and in that material. The same for LTP4, again to Valle de Santiago. Planting in March, harvesting in June. Is the distribution, and you see few lines better than Sokol and Borlaug there. And this is something I just wanted to check how is the zinc and iron of these lines, especially in the LTP4. So I sent some material to Velu and asked if we could do. And this material is basically from drought. And here x-axis is zinc, the y-axis is iron. This red line is Borlaug, and this red line is also Borlaug. And I was expecting, because Velu asked me then, can you give me the 16 lines? I don't know what is the criteria for only 16 lines. And he chose a ppm of 52 ppm, I guess. So there are 16 lines especially from the drought, with Velu probably for testing next year under yield potential and other conditions. Then we also had this year, 2019-20, the coming year, there are certain lines which is incorporated into the physiology and pre-breeding program. From yield potential, there is about 12 lines, 8 lines from drought stress, 8 lines from heat stress. And these are the list of those lines. Here is Borlaug, yielding about 6 tons. And it still has the highest 1,000 grain weight there. These lines, all of them have exotic genome in the background. You can see squarosos everywhere. Drought stress, again, the Sokol is one of the best. we still have certain lines, close to so-called. I won't say it is like really high, but still close to so-called, which goes through the pre-breeding international nurseries. Again, for heat stress, there are a few lines. Even these lines, it's lower than so-called, but we don't know the significance, how much, we didn't put that here. But it's still, if we test one more year, we can see whether it is still useful for the international nurseries. So those are the products we had until now. For the last 1 year, we are trying to put it into international nurseries for pre-breeding. But again, the seed source discovery, whenever we have an analysis, there was always a problem of the LTPs. LTPs is a group of different families. When you cross one line with another one, you get a family. You goes another one. So those families, when the previous person advanced, it had unequal numbers. So when Fernando is doing a genetic analysis, he will say there is an unequal number, so maybe we need an equal number of lines. So we had LTP 1 and 2 bulks. We went back to there. We selected equal number of lines and being multiplied in Obregon this year. But at the same time, what I did is, these bulk populations, we can also test them for yield, along with the checks. So LTP 1 and 2 bulks, I put them in the yield potential, drought, and heat, and in 2 replications, I check them. It's basically 2-meter plots, 2 reps there. And this is what I got under drought stress conditions for LTP 1. This is the whole LTP 1. You can see several lines. And here is the so-called. There is about 6 families closer or better than so-called there. And these are the, all of them has, exotics in their background. This is so-called. These are the families. And I think Johannes is very interested to know what is here. So we actually took 100 plants from each of these populations, and it's being multiplied in Obregon for further studies. Then LTP3, when I took over, there was 1 year of LTP3 data. The second year of data we generated. So this is basically the complete data under drought stress, heat stress, and irrigated optimum conditions. And I know several groups are working on it. I think Dimala is working on some of them. Johannes is working on something. So I will not go to the details. And this is what Johannes came up with yesterday. So he wanted to make genomic prediction models. And he wanted to use LTP3 as a training set. Basically, he tried 2 different scenarios. One is a cross-validation 1 scenario where you use a part of the population and predict the rest. So that basically means you are not planting the rest. You are planting only a small portion of the population, and you predict them. This is another one where we have different families in LTPs. We have, as I mentioned, there are certain families. So Johannes is basically trying to see if we can predict different families. Then LTP4, that's the newest one. We basically have 1 year of data on 695 entries, 2 replications under optimum, drought, and heat. This is the heritabilities. I just put only for the yield. And we need 1 more year of data to come up with some meaningful analysis. And future plans, because I am only 1 year in this program, what I am trying to do is to utilize the resources there and put them into a pre-breeding nursery, so this goes to international locations. And in Obregon, we are having the planting, which is going on. Certain LTPs, the bulks are being multiplied there, individual plants. And another thing which I observed in the program was there was not much phenotypic data of the parents of these LTPs, especially the exotics and elites. So those are being screened under irrigated, drought, and heat stress conditions in 2 replications. Then the best lines from LTP 1 to 4, this will be basically a product. Like from all LTPs until now, the best lines are also screened in Obregon, and then second year data of LTP 4. And the next year we are going to focus more on genetic analysis, because this year we didn't do much because I was in the process of figuring out what is happening, where is the data, etc. And I mentioned we will be screening the exotics because we have all the parents. These are genotyped already by Carolina. We are screening them under 3 environments. So this is basically how LTPs came out. All these different sets were crossed with 2 different elites. They came up into 7 different LTPs. Of these, 4 LTPs have the phenotypic data. These ones, we still don't have the phenotypic data. And these are huge numbers, like 1,000 per LTP. Instead of phenotyping them, we can actually use genomic prediction models to predict them and phenotype the subsets. And we can also validate this genomic prediction, how it works on these LTPs. That's one thing for the future. Then the second one is, this is something Johannes is working on, because when you want to say, like, there is an allele coming from a D genome, you want to know whether it is really coming from a D genome of an Agelop-Tauchi, or is it coming from a D genome of already established a hexaploid wheat. And here, if you look at it, this is a male, it's a female. If you look at this particular region, if you want to trace it back, it's not from here, it's from here, and then from here, right? But when you have multiple crosses, this particular allele formation can be observed in other, like a favorable allele, but you have to trace it back where it is really coming from. And that's what I think Johannes is working on, to come up with methods, because there are already methods in other humans and all, but we want to know really whether a faster method to apply in genome-wide association studies. The third one, this is based on a paper Sarah published. It's to identify, it's in combination with Carolina's data, basically to see, particular genomic regions adapted. When the land races are adapted to particular environments, this adaptation, has something to do with the genome also, and which part of the genome contributes to this specific adaptation. And this is an ambitious thing, might take 2 years down the road, and I think Johannes might work on it. Sarah is smiling, saying it might take more time. And the last one, this one is about development of core sets, because Carolina already mentioned she developed core sets based on diversity. Can we develop core sets based on genomic predictions? Because we have a lot of phenotypic data in different programs, and if you can use those phenotypic data, you can actually predict core sets out of the gene bank. I'm advocating for this, not only because this is my advisor from my PhD group, and I had a lot of discussions with him already, and I feel this will work. Okay. And in summary, it's basically, these are the future plans I just described. The screening of exotics, we are creating data that doesn't exist, which will help in genomic prediction, which will help in gene discovery, marker discovery, and utilizing these LTPs. Certain LTPs are multiplied. Certain LTPs, we are having more numbers. It's basically for QTL mapping and Shivas. IBD, I already mentioned, Johannes is working on. Then genomic prediction models to come up with core sets, and using those to predict the gene bank. With that, I would like to acknowledge several people, basically the Global Wheat Program and the Genetic Resources Program, and these 4 people. Without them, nothing was possible last year, because the technicians in Obregon and in Batan, and especially Cynthia, she was one of the most important person in the Seeds of Discovery program for the last 7-8 years, with a lot of contribution. And before I conclude, one more thing. This is what I have heard from several people. Like in agriculture, whatever impact a research has is in small increments. There is no magic bullets there. But we still hope there are certain magic bullets, and that leads us to look forward. But we also need small increments and also magic bullets. Thank you.
Thank you, Siva. I want to make a couple comments, but then also request everyone to, when you ask your questions, please use a microphone. Apparently, we do have some people listening in, and they couldn't hear all the questions to Carolina. So when you do ask your questions, please be sure to use a microphone. I want also to just thank you, Siva, because it wasn't easy. He's only been with our program for about a year, and he's making sense still of a huge amount of work that's been done before him by several predecessors. And what's coming together to me is quite exciting. We have plans to do some serious genetic analyses, plans that have been discussed with many of you in this room. We've had a series of meetings, and we've had lots of input, including recent proposals to CRP wheat. I think there's growing enthusiasm from colleagues across CIMMYT, and I do appreciate that very much. And we're going to find some quite exciting things from this germplasm. So I want to now open it up to questions, and please remember to use the microphone that you handle.
Thank you. Thank you very much. Nice presentation, Siva. I have two quick questions. The first one, you did not explain, or maybe, I don't know, you are new, so I didn't hear anything about the collaboration or the use of the materials that or resources generated by Carolina and group. She presented huge resources, a lot of information out there. And I believe you should be the first person to make use of those materials.
Yeah.
No? So I don't know if you have any plan or if you have already started. That's one question. The second is, how do you select the parents for pre-breeding? Because I expect you select based on the specific traits that are unique or new to the in the wheat community or in the breeding program, or the genes that are with some specific significance. So, and when you make crosses and over the generation, how do you track those traits or genes and make sure they're present at the final product?
Yeah, on the first one with Carolina, yeah, because this is the first year. Next year we'll be more focusing on the genetic analysis, and I have certain ideas. I will work with Johannes and the BSU team to come up with certain. We'll not go to the ideas now. And then the second one, how do you select parents? I mean, Matthew's program in physiology, it has been done for last 10 years or more. And I don't know if we want to change that thing or you want to come up with something new. But in this program, I haven't done any new crosses until now, right? And I was in the EAB meeting one time, and there are basically two types of pre-breeding. One where you know the traits. The one where you know the markers. And it's easier to track when you know the markers. When if you have a marker of major effect, you can know that, right? You can track them. When you don't have a marker with major effect, you basically cannot track them in every generation, especially in the early generations. So you have to still wait until probably F4 or F5 to screen them and track them. So in the physiology program, that's being done. Like, we don't track them in F2s or F3s, we track them in F5s or in F6. And then regarding how you make crosses, the simple answer to that is, if you have an elite group of germplasm, and you have a exotic group of germplasm, what you want to put there is the ones which is not present in the elites. And for that, that's what the genetic analysis basically does. The GWAS, the QTL mapping, when you compare with that, with the elite material, you want to put the germplasm that's not there, right? That's the alleles that's not present there. And that's what is basic concept of the identity by descent. We want to know whether this allele is coming from a completely exotic material or is it already present in the elite. That's basically it is. Yeah, I think Sarah.
Kind of a question and a comment. I mean, you were talking about and showed some really nice data comparing the LTPs with SOCOL and some of the other checks. I think one of the key things we need to kind of get a better grasp on is what is the allelic variation that's really driving things of value. You may have something that is yielding lower than SOCOL under heat stress, but has allelic variation that is very different to SOCOL and is beneficial for heat. So I think it's one thing and very much a breeder's perspective, which isn't wrong, to kind of just look at the material and say, okay, this is higher yielding than this one, we'll take that. But I think we also need to look back with using the genomic data as much as possible and any market trait associations we can generate to say these materials are still carrying allelic variation that is beneficial that we do not have in our elite germplasm, and then try to structure crossing patterns to try and drive the selection of that elite variation that's valuable forwards.
Yeah, I completely agree. Yeah, Matthew.
Yeah, just going. So you mentioned the two types of pre-breeding based on traits and markers, but when it was agreed that you would be in the potentially uncomfortable position of having one foot in two different programs, I think you're straddling it pretty well. I believe the idea was to combine those, right? To use some of the approaches that Carolina's using, but also the traits to get the subsets with the marker data and potentially interesting regions, and then the traits to confirm, like you were saying, that it's actually useful variation. I presume that is still the plan, right?
Yeah, that's something a discussion between you, Kevin, and Hans, basically.
I think we empowered you, but anyway, I hope. I mean, let's talk about it. I don't think Hans and Kevin are going to say that we can't. Or speak now.
Yeah, Victor.
Following up on Sarah's question, and also what you said, you wish you could do more things, but they take time. So I gather that some key constraints are number of people that you can deploy, and also testing locations. Yeah. So what would a more multi-partner, multi-location approach? What could that look like? Because it sounds like, maybe we should go down that route and build in some more people, some more locations that are comparable to Obregon, the current, plus of course the analyses, methodologies that you use.
I think it's a bigger question. Basically, Hans or Matthew has to come up with.
It's also a practical one.
Yeah,
seriously, and then we can talk about the money, but it's also practical, I think.
Yeah, in plant breeding, basically, all these experiments are trying to do is to reduce the genotype by environment interaction, and that's why we are always using multiple locations, right? Yeah, but this master group Seeds of Discovery, in the previous years, there was germplasms going out for testing in different countries, etc. But I thought, first start with a small one, and we still don't know how long the massacre is going. We have no idea. So one thing, what I was trying last year is to use, make use of what is there. And that goes to the pre-breeding nurseries of visits and satins. And that goes, we get like 100 requests from different partners around the world. So it is going there. The 4 lines from seed, it went to probably 100 locations. It's being there. But in terms of pre-breeding, if you want to really test bigger populations, let's say 200 lines in multiple locations, that has a cost.
Can I make a very short Victor,
pre-breeding is a highly specialized and long-term objectives. And you really also need to have the expertise and the people together. And if you look at wheat breeding in general, there are very, very few places where serious pre-breeding is done, and one of them is CIMMYT. Most private companies invest very little because it's a long-term commitment and the outcome is not very likely. So to say we should go out to many other places, well, we do this for screening if we screen for specific traits. But the actual breeding, I think it would not be, it would be maybe desirable, but simply not feasible to build up so many expertise in one team, which you really need to follow this carefully up. For screening, yes, and the germplasm is shared, there is no doubt, but to say that we set up multiple teams in different environments, I don't think this will be feasible.
Yeah, Velu.
Good talk, Siva. So I still wondering how Sokol can yield better than Borlaug. So we need to have a...
No, I think you need to check the data from last year in physiology and also from other programs. You can see that.
Okay,
so Sokol is basically yielding better than Borlaug in certain environments.
Yes. No, also I saw...
Even in India, if you take the data from the last year's data from the physiology trials in India, Borlaug was not yielding well because of the rust.
Yeah. No, because of some disease reaction and all this. So also I saw one of your slide. So under optimal conditions, your CV was exceptionally very high.
Yeah,
so
CV
about
was
27%. So what was the reason? Maybe because of the exotic material with the different maturity and all this?
It could be the land.
Usually we get less than 5% CV.
It could be the land. It could be the land allotted, or it could be some other errors. Because this is basically from 1 year of data. So once we have 2 years of data, the CV will go down. And also once we process the data, especially for genetic analysis, CV will go down. Yeah, Stephanie.
I was wondering, just regarding when you were comparing using leaving 100 lines out approach compared to leaving one family out, and the predictability were very low when you were using one family out. So I was wondering how you're planning to do the genomic prediction for the family 5, 6, 7.
Well, there are different approaches for genomic prediction and selection. And I don't know if Crosa is here, but normally last, let's say 10 years. Yeah, still there. Yeah, last 10 years, we was basically trying to improve the genomic prediction accuracy. That's what done last 10 years. You are using basically different types of matrices, like coefficient of parentage matrix or marker-based matrix, or even trying to put the traits like NDVI, canopy temperature into it. So you try to increase the prediction accuracy. And I was talking with Crosa yesterday, and basically that's achieved. I don't think we can really move up the prediction accuracy. What we have to do basically is to come up with strategies to use that prediction models or prediction things and come up with strategies, how you apply it, how you develop a training set, where do you apply in a breeding program or where do you apply in a pre-breeding program. That's what we are thinking. In related to your question, how to apply this genomic prediction, Johannes is coming up with basically a cross-validation 1 scenario, and that is CV1, where you use the 80% of the lines, predict 20%. There are several publications, even we have done with Crosa. This has really high prediction accuracies. There is another scenario which is called CV2, where it's basically like sparse testing, like you have missing values in multiple environments. This also has high prediction accuracy. What we are trying to do is to predict a testing set using a training set that's not phenotyped. Like this testing set is not phenotyped. The training set is phenotyped and genotyped. We have done that actually in Durham. We published in Plant Genome last 2018, where even if they are completely a little bit different sets, you can predict them. And the basic concept or idea behind this is the alleles are related. If they are completely different alleles, you will not be able to predict them. But using the exotic parents here, when we are phenotyping them and genotyping them, all of them, you can predict the LTPs. Because the LTPs come from these two populations.
So, okay. Yeah, because I was wondering if you need to phenotype those families too. Not all of them, but
no,
the 5, 6, 7. Because of the low predictability when you go across families.
No, those was done in LTP3, not on LTP 4, 5, 6, 7.
Yeah, but the predictability when you do one family leave out approach is very low. So you would assume, I guess, that maybe you need also to involve some phenotyping of those 5, 6, 7 family, because you might not be able to predict them from other families. I think that was my question.
Not from other families, but the parents of those LTPs.
Okay.
Because they are related.
Mm-hmm.
And we will phenotype them also in smaller numbers, because if you see the LTPs, they are like 1,000, so you don't want to phenotype 1,000, especially because of the variation. Yeah, Hema.
Yeah, my question comes because you use, so basically for creating these 7 LTP families or lines, you basically you selected 7 exotics and they were crossed with 2 elite lines.
7 sets.
7 sets. So because you don't have phenotypic data, how did you select?
I did.
Because for me that will, considering that there is a lot of phenotypic data on exotic material or land races or primary synthetics, if we will combine or if you will do these kind of sets, as you say at some point, based on traits or, for example, on useful genetic variation for what Carolina presented, like, okay, so let's select these lines that actually they are very different, from the elite material. I think that even the outputs, will be even, more consistent, because it seemed that it was a little bit, for me, a little bit blind. Okay, we select these lines, and after, especially under heat conditions, you have some good lines. So if it will be more directed based on genetic and phenotypic data, so.
Yeah, basically this question I cannot really answer because this was done 2011, 12, I don't know. And so I basically cannot answer how it was selected, but what I know is these elites are basically lines from Ravi's programs. There's like 50 lines, 2 sets of elites, 50 lines, best under different environments. Then elites, there are some Iranian land races, there is Mexican land races, there is synthetics. So it's basically a different, is coming up with a new design for plant breeding and pre-breeding. So there is a lot of other designs like nested association mapping, line into tester, so many different designs. North Carolina designs, 1, 2, 3. So this is basically a new design where you try to incorporate these exotics and elites, exotics into an elite background. And that's why I'm going to phenotype these exotics next year to see, the variation. Yeah, several hands there.
We need to wrap up because our time is coming to an end. I think we would welcome anyone who wants to stay back and continue discussing either with Carolina or with Siva if you have time to please do so. But I want to thank everyone for your exciting interest in these 2 topics and invite you to continue these discussions either individually or we'll have plenty of other opportunities to discuss them. So let's thank Siva and Carolina once more. And thank everyone as well for participating in these discussions. Have a nice Christmas party.